Hello, this is a video lecture for the cardiovascular system in our A&P course. So, the cardiovascular system, we're talking about the heart and the blood vessels, basically. Consists of the heart, blood vessels, and what's in the blood vessels? Blood. So you have three different components here to the cardiovascular system that we're going to discuss. So the whole purpose of the cardiovascular system is it acts as a transport system in our bodies. So it carries things around to get to various places in the body that it needs to be. So one of the primary things that it transports is oxygen and carbon dioxide. So the red blood cells are responsible for carrying oxygen or carbon dioxide and brings oxygen to the cells and then brings the carbon dioxide to the lungs to exhale or breathe that carbon dioxide out. So the blood is also responsible for transporting other types of cells. So not just the red blood cells, it's also transporting white blood cells, which are involved in immunity, platelets, which are involved in clotting. It transports around antibodies, which are involved in immunity, as well as nutrients. So things like glucose or amino acids or fatty acids. As well as it carries waste, so waste products of digestion, waste products of other body processes, and then hormones as well. So hormones that are created in one gland might need to be carried through the body to other places for it to have its effect. So the cardiovascular system is responsible for moving all that stuff around the body. So let's talk about the heart first. Heart. So the heart is basically a big muscular pump. We know that it's made up of cardiac muscle. Cardiac muscle is an involuntary muscle, which means we don't really have control over it. Interestingly, the heart will continue to beat after it is removed from a body because it does have its own impulse to keep moving, keep contracting, keep beating. So, you know, I'm sure you've seen like in movies or something where someone's heart gets ripped out of their chest or something like that. That it is accurate that the heart will continue to beat for a while after it is removed from a body. So anyways, it's a muscular pump and it has 4 chambers. So 4 chambers. And then those 4 chambers all have valves. Those valves are to prevent the backflow of blood. So we want blood always moving in a circle with the cardiovascular system. We don't want there being any backflow, okay? Because that's going to end up with backlogs, and then we're going to end up with fluid buildup in places that we don't want fluid. So typically of those 4 chambers, we have the atriums, or atria, sorry. So there's 2 atria, and then 2 ventricles. So the atria, these guys contract first, and then these ones, the ventricles, contract second. So basically, if we picture the heart, I'm trying to get my hands in here. If we picture the heart, if these are my atrium and these are my ventricles, it's like, okay, so they take turns pumping. Both the atriums, or atria, pump together, both the ventricles pump together. Okay, so that kind of covers most of the heart stuff. We remember the terms, hopefully, of systole, and diastole. So systole is when the ventricles contract, and diastole is when they relax. And then systole and diastole are both also blood pressure measurements then. So systole is the blood pressure reading when the ventricles will contract, and then diastole is the reading when the ventricles are relaxed. So let's talk about blood vessels then. So we have 3 types of blood vessels. we have arteries, we have veins, we have capillaries. So arteries are like your larger vessels. They get smaller and turn into arterioles. Same with veins, they become venules. So they have smaller versions, but if you just know the three types, arteries, veins, capillaries, I'm quite happy with that level of knowledge. So arteries are responsible for carrying blood away from the heart. Okay. So if a blood vessel is named artery, that means it's going away from the heart. Veins carry blood back to the heart. So they're returning. So I always remember that arteries are going away from the heart. And then capillaries, these are very small, like I'm talking like hair-like, tiny itty bitty little blood vessels. They're only 1 cell thick, so they're very tiny, and they are the site of gas exchange. And when we're talking gas, we're talking O2 and CO2, so oxygen and carbon dioxide. Okay, so for the most part, arteries, since they're going away from the heart and to the body, usually contain, oxygenated blood. And veins, since they're coming from the body and going back to the heart, they usually contain deoxygenated blood. So I say usually in both these situations because there are 2 exceptions, and those exceptions are the pulmonary arteries and the pulmonary veins. So if you remember, pulmonary means pertaining to the lungs, right? So the pulmonary arteries are going away from the heart to the lungs. So the lungs is where they're going to pick up oxygen and get rid of carbon dioxide. So that artery is actually going to have deoxygenated blood. It's going to be the opposite of what is usually in arteries. And then veins, the pulmonary vein is returning to the heart from the lungs. So instead of deoxygenated blood, it now has oxygenated blood in there because it's picked up oxygen in the lungs. So don't worry, I'm going to draw a diagram that I think will make it a lot more clear to you. So that's our 3 types of blood vessels. There are 4 veins that you should know. So that's the jugular. So these are all potential venipuncture sites. Potential. In a dog or cat. So the jugular, that's in the neck. Then we have the cephalic. That's in the front leg. Then I have the saphenous, which is the lateral hind leg. So lateral meaning on the side, so it's like the outside portion of the leg. And then we have the femoral, and the femoral is in the medial hind leg. In our restraint course, we get a lot more into these because you learn how to restrain 4 blood pulls from all 4 of these veins. Okay, so then we can talk about blood. So we've talked about the heart, we've talked about blood vessels, and now let's talk about blood itself. So blood has a few different components to it. So there is a fluid portion of blood. Okay, it's typically called plasma. And it's responsible for dissolving substances, so things like hormones or drugs or waste, nutrients, etc. And as well, it serves to carry the cells that are in blood. So there are a few different types of cells. We have RBCs. They're also called erythrocytes. So red blood cells, they carry O2 and CO2 with hemoglobin. So hemoglobin is the, I guess, compound you could call it, that's in red blood cells that has a great affinity to oxygen and carbon dioxide. So it carries it and then gives it up to the cells in order to let the cells have oxygen as well. Okay, so red blood cells are responsible for carrying those gases. White blood cells, these are called leukocytes. So leuc meaning white, right? Erythra meaning red, and then cytes are cells. So red cells, white cells, right? White blood cells are involved in immunity. That's about all I'm going to get into for that for now, because we cover immunity really quite more in depth in our immunity course. Okay, so I'm just going to leave it as white blood cells, immunity, and we're just going to move on for now. And then we have platelets. And platelets are involved in, they're thrombocytes. So they're involved in clotting. So thrombo means clot. So these are clot cells. Thrombocytes are clot cells or platelets. Okay. So those are the different cells that are in the blood. So I do want to talk to you about a couple different blood tests that we can run. So you guys probably have familiarity already with a CBC, which is the complete blood count. Complete blood count is looking at the cells in the blood. Okay, so for a complete blood count, we want to put a whole blood sample in a purple top tube. So different blood tubes have different anticoagulants or nothing in them. So purple top tube has an anticoagulant called EDTA. You might have seen it in some food in their ingredient list. EDTA is a common preservative and it prevents things from really like clotting together. So the anticoagulant in a purple top tube, what it does is it keeps the cells looking really very much the same as they do in just regular whole blood. So it doesn't change the way the cells look, but it prevents the blood from clotting. So anytime blood is removed from the body, if you don't add an anticoagulant in, it's going to coagulate or clot. So the purple top tube has EDTA to keep that blood in a liquid format, and it does not affect the cells. So another test that we can do is called a blood chemistry. Typically, when a doctor wants a full blood panel, they're going to want a complete blood count and a blood chemistry. So usually you're going to be pulling blood for two different types of tests. So blood chemistry, we can use two different types of samples for a blood chemistry. We don't want any blood cells in our blood chemistry sample. So we're going to pull whole blood, but we're going to need to remove the cells. So there's two different ways that we can do that. We can remove the cells and create serum. So serum is a sample with the cells and clotting agents removed. So in this case, we put a whole blood sample in a red top tube, and the red top tube doesn't have any anticoagulant in it at all. So we let it clot. And then after we let it clot, we can spin it in the centrifuge and it'll separate the clot from the serum. So we'll be able to remove the serum, which is just that liquid portion. Like when we talk back here about what's in blood, it's just this fluid portion of the blood without any of these cells and without any clotting factors in it, because all those clotting factors are caught up in the clot. Okay. We could also have a plasma sample. So a plasma sample has the cells removed. So the cells are removed, but everything else is still in there, like clotting agents and the fibrinogen, which are things that are involved in making a clot. So in this case, we're going to put a whole blood sample into a green top tube. And a green top tube contains an anticoagulant in it. It contains a coagulant that makes the cells in the blood look really strange. So we can't use this kind of sample to do a CBC because the cells will look different. We also can't use this green top tube sample to run a CBC, we can't use a purple top sample to run a blood chemistry because the anticoagulant in the green top tube affects the blood I am getting this all mixed up. So we can't use a purple top tube to run a blood chemistry because the anticoagulant in it changes some of the readings in the blood chemistry that we'd be looking for. We can't use a green top tube to do a CBC because it changes the way the cells look. Okay, so we always have to pull a purple top tube and a green top tube if we want to do a CBC and a blood chemistry. Okay, sorry, I got kind of mixed up there. So both serum and plasma contain electrolytes, proteins, drugs, hormones, and toxins. So we can get a lot of information about an animal from that blood chemistry. We can see lots of organs. If they're damaged, they're going to release enzymes that we can tell then that organ is damaged. We'll see things like creatinine and urea, which are byproducts of digestion that are usually filtered out by the kidney. If we see those in there in high numbers, we know that the kidney is just not doing their job. We can see balances of hormones and electrolytes. We can see if there are toxic levels of a toxin or a drug in the blood chemistry. So we can get a lot of information from the fluid portion of blood. So that about covers the different blood tests that we can do on an animal. Well, kind of a basic intro here. Okay, so now I want to talk to you guys about the blood flow through the heart. So you can see there's some examples in your textbook, but I honestly think they're not great, and I don't think they're awesome to study from. One of them, figure 4-11 on page 80, has a flow chart that's just words. So if you're a word learner, you might find that that is a really great tool for you for studying purposes. The other diagram in the textbook that has some blood flow through the heart diagram is 4-15b. But again, that's on page 84. Again, I don't think that's a really great diagram. So I'm going to go ahead and draw one for you here that I think is a little bit more, easy to look at and reference. So I'm just going to draw for a moment and then I will talk about what I'm drawing. Okay, I'm gonna see. I guess I can't really do a really good zoom here, but I'll kind of zoom in as best I can. Okay, so this is our diagram of the heart. So first off, your heart doesn't actually look like a heart shape. I just like to draw it with a heart shape because then we recognize that we're talking about the heart, okay? The heart is kind of almost more like fist shaped, but I like to draw it this way. Also, one of the things that you'll notice is on this diagram, so looking at my hands here, this is my left side and this is my right side. But you'll notice that this is labeled right and this is labeled left. So this is as if it is like you're looking at my heart head on. Okay, so if you were looking at my body and we're face to face, this is a diagram of my heart then. Okay, I hope that makes sense. So the right side is on this side, left side's this side. So I'm going to talk about the blood cycle, and I'm going to start in the body. So I'm going to start down here. So in the body down here, our little capillaries have given up all the oxygen that they had, the red blood cells in the capillaries, and they've picked up carbon dioxide. Okay, so now this is deoxygenated blood. There's no oxygen on those hemoglobins in the red blood cells anymore. So they're going to collect, all that blood's going to collect in the capillaries, it's going to go up the venules, it's going to go to the veins, and it's going to enter the largest vein in the body, which is the vena cava. So the vena cava goes directly into the right atrium. So the atriums are on top, the ventricles are on the bottom. If you stack them in alphabetical order, A is over V, that hopefully will help you to remember what order these guys are in. Then the blood's going to go out of the atrium and into the ventricle. So it's going to pass through what's called the tricuspid valve. Sorry, the writing's a little bit kind of tight here. It's probably hard for you to see, but you can see a little bit. I'm going to try to zoom in a bit. You can see it. I did type it up in your notes as well. So hopefully you'll be able to make a little bit better reading of it there. So it passes through the tricuspid valve. After it passes through, that tricuspid valve snaps shut, and so blood cannot get back into the right atrium then. So it passes into the right ventricle, and then the ventricles contract. It's going to push the blood out through the pulmonary valve. Again, that valve's going to snap shut so the blood can't get back into the ventricle. And it's going into the pulmonary artery. So you'll notice pulmonary valve goes into the pulmonary arteries. So remember, we said that that arteries take blood away from the heart. So blood right now is going away from the heart and it's going to the lungs. So these pulmonary arteries, I'm going to put a star next to them, are the exception to the rule that arteries have oxygenated blood. So after that blood has traveled through the pulmonary arteries to the lungs, the blood goes down to capillary level. In the capillaries, it transfers blood with the alveoli in the lungs and gives up the carbon dioxide to be exhaled and picks up the oxygen that's been inhaled. Once that blood's ready to leave the lungs, it goes through the pulmonary veins. So again, this is blood that's returning to the heart. But I'm going to put a star on that one because that's another exception. So veins usually have deoxygenated blood because they're usually coming from the body to the heart. But in this case, these veins are coming from the lungs to the heart. So they're an exception to the rule. They have oxygenated blood because the lungs is where they're picking up the oxygen. So from the pulmonary veins, the blood enters the left atrium. It's going to pass through the bicuspid valve, or also called the mitral valve. So it passes through there, those valves shut. It's in the left ventricle now. The left ventricles contract, push the blood out through the aortic valve into the aorta. The aorta is going to carry it down. So the aorta is the largest artery in the body. That aorta is going to carry the blood down into smaller and smaller vessels until it gets to the capillaries of the body. And the capillaries of the body are going to exchange that oxygen into the cells and pick up the carbon dioxide and start that cycle all over again. Okay, so I've walked you through the cycle in a big circle, but you should know that both these sides are going at once. Okay, so the atriums contract, they're going to push blood into the ventricles. The ventricles contract. They're going to push blood into either the body from the aorta or the lungs and the pulmonary arteries. At the next cycle, that blood's going to get pumped into the atrium. The atriums are going to contract into the ventricles, and etc. So both sides are going at one time. The whole thing is constantly moving. So if we track one little blood cell, it'll go around in this little circle and then back to the body, okay? But if we're talking about how things are moving, everything is moving all at once, okay? So hopefully that's pretty clear for you. You do need to know the blood flow through the heart, and you should know what parts of it have oxygenated blood and which parts have deoxygenated blood. As a general rule, if something is a artery, it's going to have oxygenated blood, because usually arteries are going away from the heart to the body. There is one artery, though, that goes away from the heart and goes to the lungs. That's the pulmonary artery, so that's our exception to the rule. It has deoxygenated blood. And then veins generally are going from the body back to the heart. So they have deoxygenated blood because all the oxygen's been used up. There is one exception though to that, and that is the vein that's coming from the lungs and going back to the heart, the pulmonary vein. That's an exception because it's coming from the lungs. That is going to be full of oxygenated blood. Okay, so those two are our exceptions. So I will tell you right now that on the test, you will not need to draw this. Okay, you will not need to label this diagram. The way you'll be tested on this on the test would be like, let's see, the vena cava goes to, or enters what chamber of the heart? And so you would say vena cava goes into the right atrium. Or when the left ventricle contracts, the blood goes into what? It goes into the aorta, okay? So you'll have kind of questions like that. Pulmonary veins enter what? The left atrium, okay? So those are the kind of questions that you would have on the test. So you should know this flow so that you know what comes next, but you won't need to draw this at all on the test, okay? So hopefully that, helps you with studying that then. So in which case that word diagram might be helpful for you if that's how you like to study. The word diagram in the textbook might be good for you then. Okay, so I just wanted to go through then our terms related to the cardiovascular system. So again, this one is going to look different than yours because it is a bit of just a messy term sheet. It has a bunch of writing all over it. It's a big mess. So feel free to consult with your typed up notes instead of this one because it'll look a little bit neater. Okay, so bradycardia. We can figure this out just from our word parts. Brady means slow. Cardia is referring to the heart. So bradycardia is a slow heart, or it's like having an abnormally slow heart rate. Tachycardia is the opposite of that. Tachy means an abnormally fast heart rate. So tachy is fast. Asystole. So we said that systole is contraction of the ventricles. So A is without or lacking. So if there's no contraction of the ventricles, there's no heart activity. So asystole means that the heart has stopped beating or a lack of heart activity. Syncope, a temporary suspension of respiration and circulation. I'm good with syncope equals fainting. Okay. Phlebotomy. Phlebotomy is the puncture of a vein to withdraw blood, but it's basically collecting blood to be able to do blood tests with. So puncture of a vein to withdraw blood. The pericardial sac. So in our little heart diagram, all around the heart, there is a big, really tough sac. It's called the pericardial sac. Peri meaning around, cardi meaning the heart, right? So it's a strong sac that's wrapped around the heart. It basically keeps the heart protected and kind of isolated from the rest of the body. An arrhythmia, that's an abnormal heart rate, right? So rhythm an A before rhythm is meaning without or lacking a rhythm. So arrhythmias can be really erratic heartbeats. So sometimes you'll have one. So a typical heartbeat has a lub and a dub sound, okay? So usual heartbeat is really steady. Lub dub, lub dub, lub dub, lub dub, lub dub. If you have an arrhythmia, it might be like lub dub, 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 lub, lub dub. So it might be all over the place. So that's an arrhythmia, where there's not really a steady beat to it. We went over these already, but thrombocytes are platelets. Thrombo meaning clot, cyte meaning cell. Erythrocytes are red blood cells. Erythro is meaning red, and cytes is cell. Leukocytes are white blood cells. Leuko meaning white, and cyte meaning cell. So CPR stands for cardiopulmonary resuscitation. So cardio referring to the heart, pulmonary referring to the lungs, and then resuscitation is trying to, basically try to get a patient's heart and lungs started again. So this is CPR. Maybe you've taken a CPR course in the past, so that you have your CPR certification. It's very similar in animals. We're going to do chest compressions to make that heart beat, and we're going to give artificial ventilation or artificial respirations to try to get some oxygen into that animal. We're going to basically be trying to save the patient's life if that heart isn't beating. Will you get to learn CPR? Yes. In veterinary nursing, we do learn CPR because it's important for all the people in the vet clinic to know how to do CPR. Okay, so edema is an accumulation of fluid in intercellular spaces, so it's kind of like a swelling. Ascites, that means fluid in the abdominal cavity. Now, I think you might be thinking right now, wait, the abdominal cavity, isn't the heart in the chest or the thorax? And yes, you are correct. So we talk about ascites in the heart section here because if the heart isn't functioning properly, we end up with fluid buildup in weird places. So, for instance, if we, sorry, I pulled the wrong diagram here. I meant to show you the one that we drew. Typically, if everything's working fine, the left ventricle contracts and pushes blood out to the body. And then there's no blood buildup because all these valves are closing behind and there's no backflows of blood. What can happen though is that these ventricles can get really weak, either really stretched out and like kind of flaccid, or really hypertrophic, which means thickened, and they're not able to send that blood out very effectively. So what can happen then is that we end up with blood back up in the ventricle. It's going to back up then into the atrium, and it's going to back up then into the lungs. So we'll see pulmonary edema or fluid in the lungs, right? Then if it still continues to back up, we're going to see that back up right into the ventricle, back into the right atrium, and then it'll keep backing up into the body. And that's where we'll see ascites. We'll see fluid build up in the abdomen. So heart disease, one of the signs of heart disease is having that fluid in the abdomen, or ascites. So pleural effusion, that's excess fluids in the tissue layers surrounding the lungs. So pulmonary edema, pleural effusion are both same names for the same thing, and that's that fluid buildup in the lungs from heart disease. So pericarditis, so let's use our word parts here. Itis, inflammation of, peri is around, cardi, the heart. That's the inflammation of that pericardial sac, or the membrane surrounding the heart. Myocarditis, itis, inflammation of, myo, muscle, cardi, heart. So myocarditis is inflammation of the heart muscle, so the muscle itself. Cardiomegaly, megaly means enlargement, cardi is the heart, so enlargement of the heart. Regurgitation or regurg is referring to that backflow of blood. So where the blood isn't effectively flowing through the body the way it should, and we see backflows. Sometimes those valves don't close properly, so we'll end up having a heart murmur, and those heart murmurs, you can actually hear the valves not closing and the blood rushing back. So when you're listening with a stethoscope or auscultating over the heart, you'll hear like a whoosh, 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 instead of a lub-dub, you'll hear whoosh, 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 whoosh. And that's that blood whooshing around instead of the blood valves closing. If a murmur is bad enough, you can feel what's called a palpable thrill. So palpable means that you can feel it, right? Palpation is feeling with the hands. And a thrill, it's kind of like a little vibration at the heart. So you can actually feel it on the animal. You touch the outside of the chest and you can feel the thrill of the blood. So hematology is the study of blood. So ology is study of, hemat is blood. So hematology, typically you're looking at the blood cells. Phagocytosis is, so phago is eat and cyto is cell. These are cells eating things. So you probably remember from like introductory bio, learning about like amoebas and stuff like that, that engulf other small creatures, etc., and eat them. So we have lots of white blood cells that are phagocytes. So they eat up things like bacteria or disease cells or debris in the body, etc. So that's phagocytosis. A hemorrhage is an excessive loss of blood. Hemostasis, so stasis is meaning stop, and hemo is the blood. So hemostasis is trying to get the blood stopping from like in a bleeding or injury situation. Lipemia, so emia is a blood condition. Lip is referring to fat. So a lipemic blood sample looks red. All blood looks red, but like the plasma or the serum is going to be red. What happened? Or sorry, I'm thinking of hemolysis. Sorry, lipemia is fat in the blood. So fat is white. So when you remove all the blood cells, so to get either serum or plasma, the serum or plasma is going to be like white, like a glass of milk. So you won't be able to see through it, and it'll have lots of white in it, and that's lipemia. Hemolysis is what I was thinking of when I was talking before. So hemolysis, the serum or the plasma is like a light pink to a red color, and that's because the red blood cell membranes have broken, and now you just have hemoglobin floating around in the blood, or in the fluid, sorry, the plasma or the serum. So what happens there is that lipemia or hemolysis can interfere with your blood results. It can be hard to read the sample properly if there's lipemia or hemolysis present. And heaven forbid, sometimes you have a hemolyzed sample that's also lipemic, and in which case it looks like a strawberry milkshake. So it's like that pink and you can't see through it at all. So there's a few things that we can do to control these things. We can control lipemia by asking for fasted samples. So if the animal hasn't had anything to eat, they're not going to have a lot of fat in the blood, and you can avoid lipemia that way. Hemolysis, we can control in a couple different ways. The first is to keep the animal calm. An animal that's flipping out and getting really upset is going to break down red blood cell membranes right inside their own body. So we want to keep the animal nice and relaxed and not stressed out. Another thing we want to do is handle blood samples really gently. So when we put our blood sample into a tube, we want to, we just invert it back and forth to let the fluid mix with the anticoagulant. We don't want to shake it. If you shake it, you can break those red blood cell membranes. We also don't want to drop it. So if a tech hands you blood tubes, be really careful to not drop them, or they're probably going to be like, "Hey, what are you doing over there?" Because we don't want hemolysis in our blood samples. So then we got a couple suffixes here that are specific to blood samples, so specific to hematology or CBCs. So sometimes we'll talk about cytosis as a suffix. So like a leukocytosis means an increase in number in white blood cells. Sometimes we have a philia. Philia also means an increase in numbers. So you'll hear about neutrophilias a lot. So neutrophilia means that there's a lot of neutrophils, which are a specific type of white blood cells. And sometimes you'll hear about apenia. Apenia means a decrease in number. Thrombocytopenia is a decreased number of platelets. So an animal with a thrombocytopenia might not be able to clot their blood properly. And then just looking at this, I think we covered all this just like in our little lecture section. Yeah, it looks like it. Okay, so that's everything I wanted to cover for the cardiovascular system. So you can read about the cardiovascular system in your Elsevier textbook on pages 79 to 84. Make sure you remember that while you're looking at those, they combine the cardiovascular system with the lymphatic system and talk about the circulatory system. We're not really necessarily concerned about the lymphatic system at this point. We'll cover that more when we're talking about immunity. So right now, just focus on the cardiovascular system itself. You can read up on chapter 9 in your McBride as well. And then I do have a review of the cardiovascular system as a practice activity for you. So you can access that and answer the questions in there as well. So if you do have any questions, make sure you do please ask during the virtual classroom, ask in the chat, or send me an email. Hopefully this has been helpful to you, and thanks for listening. Bye.